samples for the COVID cases.
How about the laboratories? Laboratory is closed?
Yeah, this I think last month, so everything had to close. So all the staff compulsory to work from home, unless you have a very critical maintenance work that you need to go to the campus. Then only you're allowed.
For 1 month lockdown, yeah.
Yeah, but still not yet lifted. So I guess probably another 2 weeks, I guess.
Yes. And same in this, our campus is now closed for 2 weeks.
Until 20, yeah.
Under 20 in July.
Oh,
but,
okay.
Limited to allow it's like in your lab. So my student is
yeah, a lot of our students' work has been affected, right? So
yes.
same here. So they can't go to the lab and do experiments. So you know, their progress definitely will be you know, delay.
Yes.
Yeah. Yeah.
So, Dr. Tan Buncin, we will start in around 10 minutes.
Sure, no problem.
Yeah. How should I address you?
You can just call me Boon if you want, just in short.
Sure. Yeah.
Also can, no problem.
Yeah, thank you.
Bu Fatma belum hadir ya Pak Anja?
Telepon ya?
Ya, belum.
Sebentar. Selamat pagi Bu Fatma. Oh iya, terima kasih. Terima kasih.
Selamat pagi. Assalamualaikum. Sebentar saya ganti ini dulu. Apa?
Virtual background.
Yeah.
Gimana Pak Yasin, Bu Fatma? Baik, sudah?
Oke, kondisinya sudah membaik. Memang ini sih sebetulnya beliaunya baik. Cuman masih menunggu itu hasil yang tes yang kedua sudah hari Senin sebetulnya. Cuman karena antri mungkin jadi lama.
Iya betul. Antri ya.
Hasilnya, okay.
Betul.
Sehat semua Pak. Siapa Pak Saifu masih di IGD katanya.
Katanya sudah pulang.
Kemarin
iya,
bukannya ada.
Karena bukan COVID.
Karena bukan COVID.
Oh, enggak tahu info terakhir kok sudah di IG sudah di ICU, masih di ICU kok. Kemarin info dari Bu Mira kan.
Jam berapa?
Sebentar. Jam 9:30 malam.
Oh.
Jam berapa?
Soalnya kemarin ada berita katanya disuruh pulang karena bukan COVID.
Ini dari Bu Mira dapat WA dari siapa? Prof Nyoman katanya sudah di VIP lantai 7B. Jawaban Prof Nasron. Tapi kata putri Pak Saihu, Pak Sah masih di ICU. Itu jam 9:30.
Oh.
Oke, mudah-mudahan
soalnya sore itu sudah nggak on lagi, maghrib.
Oke, mudah-mudahan kondisinya memang segera membaik.
Mudah-mudahan lah.
Agak mengkhawatirkan.
Ya.
Loh,
Ini
Bu
juga
Fatma, Pak Eri itu juga kena kah?
buatan kemarin yang
Oh,
waktu
enggak toh.
tes swab antigen negatif sih.
Oh ya sudah. Soalnya simpang siur ini loh beritanya.
Karena
Mudah-mudahan enggak semua lah.
Nggih, tes kok Pak Eri tes memang nggak bisa.
Tapi kemudian Pak Eri itu kan tetangganya kena, kemudian diisolasi. Tapi hasilnya sudah negatif. Nggih.
Tes kok Pak Eri tes memang nggak bisa.
Tapi kemudian Pak Eri itu kan tetangganya kena, kemudian diisolasi.
Reagen ya untuk PCR-nya.
Okay, so we will start now. Assalamualaikum warahmatullahi wabarakatuh. Selamat pagi, good morning everyone. Welcome to the second seminar on tropical plant biotechnology. This seminar series is held every 3 to 4 months by Biotechnology of Tropical Medicinal Plants Research Group, Universitas Airlangga. The official name in Indonesian is Group Riset Bioteknologi Tanaman Obat Tropis, abbreviated as Biotanotrop. So I'm Anjar Triwibowo. I am lecturer and researcher from Department of Biology, Faculty of Science and Technology, Universitas Airlangga, and member of BIOTECNOLOGY. And I will be the moderator and facilitator for our seminar today. Before we start, on behalf of the faculty and university, I would like to give our utmost appreciation to our honorable guests, the Vice Dean of Faculty of Science and Technology, Universitas Airlangga, Dr. Fatmawati. Thank you for joining us today. Also, the head of BIOTECNOLOGY, Professor Wulan Manuhara. Thank you for joining us today. I also would like to give our huge appreciation for both of our speakers, Dr. Nani Siti Aminah and Dr. Tan Bonchin, who are willing to allocate their time to share their work and research with all of us here. So thank you very much for our speakers. Okay, I also want to give our huge welcome and appreciation to all of our participants from all over Indonesia and the world. Today we have participants that came from different countries. We have mostly from Indonesia. We also have participants from Malaysia.
Recording in progress.
We also have participants from Malaysia, Pakistan, Myanmar, Nigeria, Iran, China, and Bangladesh. Thank you, everyone, for joining us today. I hear that in Nigeria right now is 3 a.m. in the morning, but some of you joining us today, so we really appreciate that you joining us in the middle of the night. Thank you. And especially thank you everyone for joining us in this difficult time in the middle of the COVID-19 pandemic in Indonesia. So for you that not coming from Indonesia, we have the second wave in Indonesia. We have more than 30,000 cases every day, and the healthcare service is a bit overwhelmed, is kind of overwhelmed. So thank you so much everyone for being here today in the middle of this difficult time. Hopefully this seminar can positively contribute to overcome this ongoing pandemic. Amin. Okay, without further ado, we welcome the Vice Dean of Faculty of Science and Technology, Dr. Fatmawati, to give the opening remarks. Dr. Fatmawati, the time is yours.
Okay, thank you, Pak Anjar. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Honorable Speaker, Dr. Tan Bonchin from University of Malaya, Dr. Nani Siti Aminah, Department of Chemistry, Universitas Airlangga, and also Professor Dr. Sri Wulan Manuhara, Head of the Biotechnology of Tropical Medicinal Plant Research Group, lecturer, student, and all participants. Good morning, ladies and gentlemen. I am pleased to welcome you to the second seminar series on tropical plant biotechnology. It is high time that such an event took place to highlight the remarkable contribution which tropical plant biotechnology make in so many areas of our life, and we all understand that it is the ways in which they are transforming the world. Was not clear earlier. It is blindingly obvious now in the time of the COVID-19 pandemic and still in progress. Science, technology, and innovation are indispensable for our response to and recovery from this crisis. This online conference is our opportunity to demonstrate that transformation is possible and is happening right now in so many places with so many innovation and with engagement of so many people. Biotechnology is embedded is the Sustainable Development Goal together with advancing science and technology as goal 14 and 15. However, life science and biotechnology must not be limited to SDGs 14 and 15. They cut across all the SDGs as an important element of implementation. Ladies and gentlemen, I believe that by participating in this online seminar, we are in the right place and the right time. Together, let us accelerate the exchange of ideas and scaling up of good practices. I'm confident in this event, you will find new ideas, fresh energy, and novel partnership to sustain your effort in support of the SDGs and recovery from COVID-19 through life science and biotechnology. I wish you every success with this important conference. I look forward to learning about the outcome. Thank you. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you very much, Dr. Fatmawati, for the opening remarks. So now we have opening remarks from the head of Biotechnology Research Group, Professor Wulan Manohara. The time is yours. Thank you.
Thank you, Dr. Anjar. Good morning, everybody. First of all, I would like to thank Dr. Fatmawati, who has been pleased to open the second seminar series on tropical plant biotechnology. I also thank and appreciate to Dr. Nanik Siti Aminah and Dr. Tan Buncin, who has agreed to be a speaker at today's seminar, webinar. Thank you so much for all the webinar participants for attending this second webinar. Hopefully that the topic from the speaker at today's can be useful to increase our knowledge. On this occasion, I will launch our group research website, namely https biotanotrop.fst.unair.ac.id. Maybe I want to share screen. Okay, my screen is
Yes, you can see the screen.
See the screen? Okay.
Yeah.
This is our website, namely biotanotrop.fst.unair.ac.id. Now the website still available in Indonesia, in Bahasa Indonesia, but in the near future we will make it in English. Yeah. So this about us, the vision, mission, our vision and our mission, and then we have 8 members. Me as and Prof. Heri Purnabasuki, and then Dr. Alvinda Novi Kristanti, and Dr. Anjar Triwibowo, and Dr. Nanik Siti Aminah, and Dwi Kusuma Wahyuni, SSI MSI, and Dr. Sugiarto MSI, and Armando Gerardi SSI PhD. And we have 4 collaborator research. This is Professor Iqbal Kodari, from International Center for Chemistry and Biology Science, Pakistan, and Professor Yoshiaki Takaya from Faculty of Pharmacy, Meiju University, Nagoya, Japan, and Dr. Tan Buncin from Center for Research and Biotechnology for Agriculture, University Malaya, Malaysia, and Dr. Mugunda Rauf from Faculty Science, University of Malaya, Malaysia. Yeah. This is our research. We have many research from 3 years ago. Is from 2020 until now, and 2021 and 2020 and 2019 and 2018. We also have facilities in this our laboratory in Faculty Science and Technology, Universitas Airlangga. We have many kind of equipment to support our research. And then we have also published article for 4 years ago. We can see in this menu. Yeah. So in the end of this web, if for participants who want to collaborate, please contact us in the contact menu. Yeah, we can maybe the participant of this webinar want to collaborate with our research group, please contact us in this menu. We hope all participants can join with our research so we can explore the natural product from Indonesia or in a tropical country to be increase the research about the tropical medicine. Thank you, Dr. Anjar. I think it's enough for the launch of our website.
Okay, thank you very much, Professor Wulan Manohara, for the remarks and for introducing, for launching and introducing our website. So our website, I would like to remind you again that our website it's biotanotrop.fst.unair.ac.id. You can find our website. We post it in the chat feature of Zoom. So we will be really happy if you all can visit our website, see our research, our publication, and our facility. And we would like, we will very welcome you all to joining us to collaborate in research and in education, or if you want to send your student to Biotanotrop, to Faculty of Science and Technology for internship and everything, we will be really happy. So please visit our website and contact us for potential collaboration. Thank you. Okay, so now I would like to introduce our first speaker, Dr. Nani Siti Aminah. Dr. Nani Siti Aminah is the member of Biotechnology of Tropical Medicinal Plant Research Group. She is Associate Professor and Researcher at Department of Chemistry, Universitas Airlangga. She finished her bachelor from Universitas Airlangga, then her master and doctorate from Bandung Institute of Technology, Indonesia. She has working experience as postdoctoral researcher and research fellow at Meiji University, Japan. She has expertise in natural product chemistry, and she has published more than 50 papers and book chapters in this topic. Today, Dr. Nani Siti Aminah will give a talk about conventional chromatographic techniques for isolation of phenolic compounds. Dr. Nani, we look forward to hear your talk, and the time is yours. Thank you.
Thank you, Dr. Anjar. May I share my, can you hear my voice?
Yes.
Okay.
Hear you.
Okay. I will share first my slide. Okay. Yeah. Can you see my slide clearly?
Yes, we can see it.
Thank you. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh, and good morning. First of all, I would like to say thank you very much to Dr. Fatma as Vice Dean on Research and Partnership in Faculty of Science and Technology, Dr. Tan Buncin, Dr. Professor Wulan Manuhara, and all of the members of Biotechnology of Tropical Medicinal Plant, all of the professor, colleagues, lecturer, student from Universitas Airlangga, from the outside of Universitas Airlangga in Indonesia, Malaysia, Pakistan, Iran, Bangladesh, Myanmar, and China. On the first slide, I would like to introduce again, after Professor Wulan introduced all of us, here you can see how handsome and beautiful we are. So you can see also all of our field in our work every day. Okay, by looking this picture, you can see we are all work in the natural product chemistry and in biology. So that's why we work together to get some result, that good result. Hopefully that we will get so many funding in the future. As part of a biotechnology of tropical medicinal plant research group, I feel proud and very grateful to have the opportunity to deliver today's presentation. Even though the current situation in Indonesia and also in our city, Surabaya, very unfavorable. Our laboratorium now also closed, but all of our group in this research group is still eager to continue sharing our knowledge. So let us pray together, hope this situation will get better and better. On this occasion, I would like to present I would like to share our experience about conventional chromatography technique for isolation phenolic compound. This is our experience that we have worked this part start from 2000. Okay, before we go to the chromatography, I think I want to introduce our country because I believe that some of you that joined today didn't know where is Indonesia. But I think also all of you know where about Bali. So I want to introduce you Indonesia. This is Indonesia area, so huge place. And then we are Surabaya in the part here in the east of Java that near from Bali Island. I want to show you that Bali Island is a small island part of Indonesia, but very famous in the world. When we go to the overseas, sometimes if then they want to know where you come from, we say that we are from Indonesia. Where is Indonesia? But if we say that we are from Bali Island, oh no, I know Bali. So I want to show you that Bali is a small island near our island, Java Island. Okay. When you visit to our city in Surabaya, you have to meet with two statue. Yeah, this is the bambu runcing, that sharp bamboo, and then also the Surabaya statue. That when Professor Takaya and Professor Nintanda Aung visit to our city Surabaya, we bring together for them to go to this place. We also from Surabaya, maybe 2 or 2.5 hours, we can visit the beautiful mountain, the most beautiful mountain in the morning in the world, I think. We call the mountain is Bromo Mountain. Not far from the Bromo Mountain, we can go to the lake. This is very peaceful lake that you can contemplation in this place to get some idea for your research. I think that we promise if you visit to our country, we will bring you to these two places. This very beautiful. Okay, I want to show you our university, Universitas Airlangga. Universitas Airlangga have 3 campuses. The campus A for the medical. This is for medicinal faculty and veterinary, and dentistry faculty. And then B campus, there are the social science faculty in here. And then in C campus, when our faculty, that is the C campus, that's very beautiful in the night, that our faculty of science and technology belong to the C campus. So that are the Surabaya, Indonesia, and also Universitas Airlangga. So now we go to our main discussion today. What is the phenolic compound? The phenolic compound is a class of chemical compound in organic chemistry which consists of hydroxyl group directly bonded to an aromatic hydrocarbon. All other members of phenolic family are known as derivative of phenol. That what is chromatography? The chromatography first derived from the Greek chroma, meaning color, and graphene, meaning to write. The technique was originally developed by the botanist from the name is Sweat. The analytical technique utilized from the separation, purification, identification of constituents from the mixture. It worked on the principle of differential interaction of solution with different phase, the stationary phase and mobile phase. Many modifications were made to the technique of chromatography to overcome the shortcoming like analysis time and the range of the compound that could be detected. Application of pressure were practiced by use of pump to reduce the time of run. So I want to show you the video about the basic of the chromatography. Let's enjoy to see the video.
Welcome to a brief introduction to the technique and principles behind thin layer chromatography. I'm Professor Davis from ChemSurvival.com and the YouTube channel ChemSurvival. Let's get started. Now, you can't really have a conversation about chromatography without first having a conversation about Mikhail Svet. Svet was a researcher working in Russia on his second PhD when he made a very interesting observation. He was working with plant pigments, and he discovered that he could dissolve many plant pigments in relatively low polarity organic solvents. But he noted that when he added one more thing to the system, things changed. He added insoluble plant material like cellulose fibers. And when he did that, he noted that the solubility of the compounds that he was working with changed drastically. This is a very interesting observation. He could affect the solubility of compounds simply by adding an insoluble material to the solution. This was a very curious observation. And it may not seem obvious now, but in just a moment, I'm going to explain to you how Svet turned this into the most powerful separation technique that chemists have in their entire arsenal. So Svet had made a very interesting observation that solutes tend to jump back and forth between an insoluble solid phase and a liquid phase above it. But the real utility of his observation comes when we introduce a second compound to the equation. Recall that when he added a high polarity compound to a solvent with plant material in it, like cellulose that's insoluble, that high polarity compound tended to stick very strongly to that material and not be dissolved very much. But if we introduce a compound of lower polarity, it has less of an affinity for that polar compound, and therefore will spend more of its time dissolved in solution. But notice that these are equilibrium processes. We call this equilibrium partitioning, and it has to do with how strongly a compound adheres to a solid phase versus how well it dissolves in the liquid phase. In our case here, the high polarity compound favors the solid phase, whereas the low polarity compound favors the liquid phase. But how do we translate this into a separation technique? The answer to that question is we have to get that liquid phase in motion. So instead of having just a pool of liquids sitting on top of a solid, let's instead have that liquid moving across the surface of the solid. We're going to call that flowing liquid our mobile phase, and we're going to call that stationary solid our stationary phase. Now let's add that high polarity compound to the system and see how it behaves. Remember that the high polarity compound spends much of its time adhered to the silica and very little of its time dissolving into that flowing mobile phase. So when we release this compound and let nature take its course, notice that it spends a great deal of its time stuck to the silica and therefore is moving very, very slowly across the screen. So slowly that I have to say something else just to keep up with the time. Done. Now, let's change that. Let's instead add a low polarity compound. Remember, the lower polarity compound spent a much greater deal of its time dissolved in the liquid phase and less of its time adhered to the silica. So when I release this low polarity compound and let nature take its course, let's see what happens. Notice it's moving much more rapidly across that stationary phase than its more polar counterpart did. So now let's add a mixture of both to the same point on that plate. And again, release it and let nature take its course. What do we expect to see happen here? Well, of course, they move at different rates, and therefore they become separated in space very quickly from one another. This is the essential principle on which chromatography relies. Compounds with greater affinity for the stationary phase will move more slowly than compounds with greater affinity for the mobile phase. And therefore, if we have them all start in the same location, they're going to end up in different locations. And that, my friends, is the definition of separation. If you're taking an organic chemistry course for the first time, your first exposure to chromatography will almost certainly be this, TLC, or thin layer chromatography. In this technique, we use a glass or plastic-backed plate covered in a very thin layer of silica, which is essentially very, very tiny glass beads. So it's a highly polar surface. But how exactly does that surface help us to separate compounds in the lab? Well, we have to take a few steps, obviously, to make this happen. The first is we need to mark our starting lane so we know exactly where we're placing our samples when they begin. We do this using pencil. because many inks actually have organic compounds in them as pigments, and those organic pigments will chromatograph with everything else. We don't want that to be part of our experiment, of course. We mark it about 1 centimeter above the bottom of the plate, so that when we dip the plate into a shallow pool of mobile phase, we don't submerge the spots. We've got to be sure that it's higher than the mobile phase pool, because if it's not, then the chromatography experiment is going to fail. We have to be sure that our compound only has 2 options, either dissolve into migrating mobile phase or stay adhered to the plate. Dissolving into the mobile phase pool can't be an option. The next step in the process is to apply our compounds to the plate. Now, this is often done using small glass capillaries, which we can fill with solutions of our compounds in a volatile solvent, such as methanol. We use open-ended capillaries so that we can draw the liquid in through capillary action and also expel it from the capillary simply by touching it to the surface of the silica plate. So I'm going to spot one compound here on the left side of my plate. I'll spot my other compound here in the middle of the plate. And just for demonstration purposes, I'm going to perform what's known as a spike, which is I'm going to spot a mixture of both solutions on the plate as well. So now I have very thin layers or very small spots of my compound on the thin layer of silica. These spots have to be very small. The smaller they are, the better they are. You can make them more concentrated by tapping the capillary to them several times and allowing the spot to dry in between those. It's important that they be at least 1 full spot diameter apart from one another and away from the edge of the plate. This is because as the chromatography experiment progresses, these spots naturally tend to expand a little bit because of diffusion. Now we're about ready to run our chromatography experiment. We're ready to do what's called developing the plate. And to develop the plate, we submerge just the very bottom of the plate in a very thin pool of our liquid mobile phase. When we do this, that liquid mobile phase will begin to migrate up the plate, just like water being sucked up into a paper towel that's been dipped into a pool of water. As that flowing mobile phase moves up the plate, it will encounter the spots. And at this point, the spots have a choice. They can spend more of their time dissolved in the flowing mobile phase, or more of their time adhered to the highly polar silica stationary phase. And as this happens, the spots migrate. Now notice that I've stopped my plate with the mobile phase about 3/4 of the way to the top. I can't let my mobile phase reach the top, as we're about to see, because there's a very important calculation I need to perform. At this point, my plate is finished developing. I have to quickly mark that solvent front, because if I don't mark it rapidly, that solvent will eventually evaporate, and it's very difficult to detect where it was at all. So now I have a developed TLC plate, and I can clearly see that my yellow compound has moved farther than my purple compound. So my yellow compound must be less polar. But we prefer to have a more quantitative way of describing how these compounds move.
Okay, I will continue my presentation. Why I show this video, because this is the basic of our work in our lab. So I want to show you the type of chromatography. In this presentation, I divide to become 2 parts. First one is the conventional chromatography that already we always use in our lab. The first one is the thin layer chromatography that already explained in the video. And then the second is the paper chromatography. The third one is vacuum liquid chromatography. And the last one is gravity column chromatography. The second part of the chromatography is the sophisticated chromatography. They are radial chromatography, gas chromatography, and then HPLC, GCMS, and LCMS-MS. It is shown in this here, in this slide. This is very expensive. But today I don't want to explain every single of this chromatography, but I want to share our experience how to isolate the phenolic compound from the sample to get the pure compound. Okay, strategy to isolate the phenolic compound, we start from extraction process. That we usually have the sample that in powder form, and then the powder form, then we do extraction. That you can see in this picture that extraction process also very simple. I think all of you have the jar in your house, that usually in your jar there is the crackers or something like that. But in our lab, we use the jar to do extraction using maceration process. After we do extraction, and then we filter our extract and then evaporate by rotary vacuum evaporator, and then we get the extract. After we getting the extract, so we continue separation using liquid process, that we call it separation by liquid process, that we can remove the non-polar compound using the hexane, and then we continue the separation using the same process by liquid extraction, that we can get the semi-polar compound that we believe that the semi-polar compound will get in this fraction. So the semi-polar fraction then separate using conventional chromatography. We usually first use vacuum liquid chromatography, and then we continue using gravity column chromatography until getting the pure compound, and then continue by spectroscopy method to analyze, and then the bioactivity, and finally we get the phenolic compound structure and also the bioactivity data. How we separate the compound using chromatography? What we do in our lab for the first time. Usually, after the student get the extract, after extraction process, all of the students try to get the profiling using TLC. They do many type of ratio of the solvent. This is the star anise extract that our students try to make many type of ratio of the solvent. You can see here that hexane dichloromethan 9,1, hexane dichloromethan 7,3, and so on until you can see here, if we use the solvent that non-polar, only one spot that move, as we can see from the video, that after we increase the polarity of the solvent, the second spot is moving also. It means if we separate using vacuum liquid chromatography and also gravity column chromatography, they will go down first. Okay, we also try to use another ratio of the solvent using hexane ethyl acetate, and then we can get the picture like this. So from this process, from this profiling, usually the new student that come to our lab, I and Ibu Alvinda will give them the formula how to separate for the first time. That look like the medical doctor that gave the recipe for our patient, and then here is your recipe here or your recipe, and then after that with this recipe they try to separate the compound using vacuum liquid chromatography. Why we choose the vacuum liquid chromatography for the first time? Because with vacuum liquid chromatography we can do the amount of the sample 30 until 40 grams of sample, and then the time running also fast because of the vacuum. If we choose the good solvent, if we choose the good solvent, we can see here that 1, 2, 3, 4, 5, 6, 7, maybe 10 of compound can separately very good. This is the one of the species, I think, star anise, and then is it the other species is this one also the other species, the result of vacuum liquid chromatography identified by TLC. And then after that, we will choose which one of the spot that they are same, we can combine and we call it subfraction. After we get the subfraction, we do TLC again to find the solvent that will use for the next chromatography, because now our subfraction is the amount become a liter. Usually we use the gravity column chromatography. If we choose a good solvent, we can see clearly that that our separation becomes so beautiful. You can see here in our column that the first compound will go down first, and then we put in the bottle, and then the second compound will come after, and then the third compound will come after. So we can separate very, very good to get the pure compound. We can see here also that how to separate our compound here. This is the result of vacuum liquid chromatography. And then we took this part for separate again. Of course, another part also separate because we don't want to one of the compound, we want to all of the compound can separate well to get the pure compound. So from this part, we separate again, and then separate again until we found the pure compound with spot, only one spot. And then after we get the one spot, we check again by TLC with 3 kind of solvent until we get the one spot only. So after that, we do analysis using spectroscopy. And then in this spot, we conclude that the structure of the compound is like this. Yes, the dimer of stilbenoid that we call Diptoindonesian A, because at that time, this is from Dipterocarpaceae, the first time isolated from Indonesia. That's why we gave name Diptoindonesian A. So after that, we with the same process, we get maybe more than 100 compound, secondary metabolite compound from plant, also from synthesis. Here I will show you how all of the compound can get one by one. Here are the result of vacuum liquid chromatography. And after the process, we can get this part is this compound, and then this part is this compound. This part is this compound, this part, and all of the part can separate well. Here are all the structure of the compound that isolated from Clausena excavata only with vacuum liquid chromatography, gravity column chromatography, and TLC. That's all this compound already found by Dr. Tin Myotan, our student from Myanmar. Here are another one is from Cassiniora edulis. There are flavonoid compound already isolated, also coumarin and granulated of coumarin. They separate also using 3 types of chromatography, 3 types of conventional chromatography. So another one, we try also separation of stem bark of Cassia multijuga. There are type of compound of anthraquinone, the dimer of anthraquinone. This compound color is very, very beautiful, a yellow color. That's very interesting compound. So another one, we also separate from Cassia javanica. We found from this species flavonoid compound and also anthraquinone compound. And another one, our group research also worked with the grape that we found the terpene compound that this compound name is resveratrol. I think if you Googling that you write resveratrol is very expensive price of this resveratrol. You can find from the seed of the grape, the grape that is very cheap in Indonesia because the taste is sour, so no one people that like to eat it. That if you want to get the resveratrol gira. So we also found the epsilon viniferin, the dimer of resveratrol from this species. We also work with Sorea seminis. That from this species, we found the oligostilbenoid compound from monomer until tetramer. So the other species of Sorea, we isolate the similar compound. The interesting of this compound is the dimer compound, the dimer stilbenoid compound containing vitamin C in here. We also found the tetramer, trimer, dimer. So we go to another family. This is Anunaceae that our work last year, Bu Alvinda. This we found from this species from Sarcopetalum, we found the flavonoid compound. That's very interesting also. And then we continue also our work in yam. That's, I think, Indonesia people all know. The Myanmar people also know this. We usually eat, last year, we usually eat for breakfast, this one. There is two compounds that very interesting compound with the activity of antioxidant also very high. And then we also work with all of the compound of the plant from Indonesia. And then we also try to synthesize of the compound, the major compound, we also synthesize to be the derivative of the compound. From all of our work in chromatography, we conclude that the first pure derivative phenolic compound can be isolated from extract using conventional chromatography technique. So if you don't have in your lab the sophisticated equipment, don't worry, you can get the pure compound. So the second one, the TLC analysis is the main reference in doing separation in determination eluent for separation and purification by PLC and gravity column chromatography. And the last, I think this one is very important, initial screening with TLC, the patient of the student that work in the lab, thoroughness, strategy, and careful preparation are the key to success in process of isolating phenolic compound. So here are our publication, a part of our publication. And then maybe this is a part of our student that work together in our lab. We are always happy after go out from the lab. But if we and all our students work in the lab, we are so serious. But if we are go out from the lab, we are so very happy. This is the graduation of our student. So thank you very much for all of your attention. Thank you very much, Dr. Anjar.
Thank you very much, Dr. Nani Siti Aminah, for the wonderful presentation and talks. So before we go to the second talk, I would like to remind all the participants, if you have any questions, you can write it down in the chat feature of the Zoom, and we will read your questions during the discussion sessions. Or during the discussion, you can also ask your questions directly to our speakers. Okay. So now, without further ado, we go to our second speaker, Dr. Tan Bonchin or Dr. Boon. So Dr. Boon is a researcher and lecturer at the Center for Research in Biotechnology for Agriculture or CBAR at the University of Malaya, Malaysia. He completed his bachelor degree in biotechnology in University of Putra, Malaysia or UPM, and a PhD in plant biotechnology at the University of Nottingham, United Kingdom. His research work and interests including the area of plant molecular biology, plant biotechnology, and biochemistry. He published more than 30 papers, book chapters, and patents in those topics, and receiving several prestigious awards and recognition for his work in the topic. So, Dr. Boon, we are very excited to hear your talk. The time is yours. Thank you.
Yeah, thank you, Dr. Anja. Let me share my screen first. Right, so I believe you all can see my screen and also hear me.
Yes, we can hear you clearly and we see the presentation.
Yeah, thank you very much. So very good morning to all from Indonesia and also from other countries. First of all, I would like to thank Prof. Wulans and also the organizer for giving me this opportunity to share with you some of the research works that have been done in our labs. So today I'm going to talk about metabolic engineering of gingers for the productions of flavonoids. My name is Bonchin. I'm from University of Malaya, Malaysia. So the gingers we are going to talk about is the Bosombagia rotunda. So this is one of the medicinal gingers. So if you look at the image in the middle here, so this is how the Bosombagia rotunda's plants look like. So it can grow taller than the tables at the back here. So the red circle here is the rhizome. So if you zoom in, so you can see this is how it looks like for the rhizome. So it's quite similar to our finger. So that's why we call it finger root ginger in English. And in Malay and also in the Indonesian language, it is also known as temukunci. So because this shape also looks like, you know, the bunch of keys, right? So Bosombagia rotunda is a common edible ingredients in many Asian countries. So it belongs to the families of Gingiberas, so which is the ginger family. So it is commonly grown in Southeast Asia like Indonesia, Thailand, and also Malaysia, as well as India, Sri Lanka, and also southern China. So people usually use fresh or dry gingers in cooking, and some take ginger supplements for their possible health benefits. So this is because gingers contains a lot of bioactive compound, and of these, flavonoids are the major constituents. So in the past, many bioactive compounds have been isolated from Bosombagia rotunda, and it has been shown to possess a variety of biochemical activity, such as antioxidants, anti-inflammatories, and anti-carcinogenics, and etc. So there's another group called prenulated flavonoids. So this is also one of the natural flavonoids that have, you know, the prenulated side chain attached to its skeletons. So it has been reported that these prenulated flavonoids are generally more bioactive compared to their corresponding flavonoids. So here is an example, naringenin. So this is also one type of flavonoids. When there's a prenyl group here attached to its carbon 8 at the positions number 8, then it will become a 8 prenyl naringenins. So this process called prenulations. Now, since we know Okay, so since we know that gingers contains a lot of useful flavonoids, and so we started to like, okay, so maybe we can analyze all the organs and then to identify which organs in plants actually contains high amount of, some of the selected flavonoids. So as expected, if you look at the table 1, so we found that the areas parts of the B rotunda contains a highest amount of flavonoids tested compared to non-areals parts. So among the selected 8 flavonoids, so pinostrobins was the highest compound detected in rhizome, and followed by pinostrobins compound. So these 8 compounds are selected flavonoids can be categorized into chalcones, the first group, or the second group flavonones, and also the third group cyclohexanils chalcones, which in short I will just call it CCD. Then we check the inhibitory activities of these flavonoids against dengue 2 virus and S3 protease, and we found that among these 6 selected flavonoids, pandrotin A and 4-hydroxypandrotin A, which is belongs to CCD, the cyclohexyl chalcones groups, produce the highest percentage of inhibitions against the dengue 2 virus. So we are quite excited because we found something, the potential compound that have anti-dengue property, which is important for our, the tropical country especially. Then we wonder, like, how this compound actually biosynthesized. So we check on the flavonoids biosynthetic pathway, and we know that flavonoids biosynthesis is starting from phenylalanine, this compound. So this is the first precursor. Then, but we are not sure how from these phenylalanines then it can develop or produce at the end the cyclohexyl chalcones, which is the CCD compound here. So like we sought help from our collaborators, chemists, to see like, what are those compounds and then how this compound actually can, produce from this plant, because we are not very familiar with these chemical structures of the compound. Of course, when you ask a chemist on how these compounds was produced, everything, and how they can get it, they will just, we synthesize everything. So in fact, they successfully synthesized the CCD, which is pandrotin A and 4-hydroxypandrotin A, using a chemical way. But of course, believe me, I do not really know how this process works. But what I do know is this process is quite expensive and not really cost effective. So we, as a biologist person, so I believe we also can do some things, right? Besides synthesize the compound, and to understand how this compound being biosynthesized. So for example, one of the strategy would be the manipulating the CCD biosynthetic pathway in this Bosombegia rotunda plants. So there are 3 major approaches that we actually can use to enhance the productions of CCD. So the first strategies is through upstream precursor overexpressions, where you have overexpress this particular process so that at the end you can get the product that you want. Like for example, in this case, if you want to get the product number 3, so you can do upstream precursors overexpressions here, so that hopefully at the end, throughout the whole process, you can get a higher amount of this product number 3. So we try this strategy first in our case. So if you look at the flavonoid biosynthetic pathway again, so again, the first precursor would be phenylalanine. So we was thinking like, okay, maybe we can using this upstream precursor overexpression by supplying more phenylalanine compound to the B rotunda cells. So to do this, of course, we have to first establish a cell suspensions culture first. Okay, so from the rhizome, okay, so we try to induce the shoot from there. If you want, you can actually from cell suspension culture regenerate back into the whole plants. But in order to study the genetic engineering works, we do not go until a whole plant. We stop at the cell suspensions culture, and then we use these cell suspensions cultures for metabolic engineering works. So since we have a B rotunda cell suspensions culture, so we try to add in the alanines compounds into the cultures. And then, of course, we tested different concentrations. So in general, we found that 40 milligram per liters phenylalanines actually can enhance the productions of the selected flavonoid here compared to the control, which is untreated cell with this phenylalanine. Then after treating the cell, so we harvested the cell suspension cultures. Then we analyze using the transcriptome approach, which in this case we are using RNA sequencing. So after analyze all the data, we found that after phenylalanine treatments, so several unique genes related to flavonoids biosynthetic pathway were upregulated. So in total, actually, there are 14,644,000 unique genes were upregulated in red color, and in green colors is 14,379,000 unique genes were downregulated in this case. So of course, they can be classified according to their functions. And then we verified using a qPCR for some of the selected flavonoids related genes to check whether is there tally with our RNA sequencing data. So besides looking at the RNase level, we also look at so before that, so we mapped back to this biosynthetic pathway, the flavonoid biosynthetic pathway. We found that those genes related to this process were actually upregulated. Okay, so besides looking at the RNase level, we also look at the proteomes level, which in this case, we are using the gel-based proteomics approach. So this was quite a long time ago. That's why we are using gel-based proteomics rather than the gel-free, because at that time, the gel-free is not so common and we do not have machines in our lab. So we are using a gel-based. So we run the two-dimensional gel electrophoresis to separate the proteins from the phenylalanine treated cell. Then we found that out of 34 proteins out of 76 were successfully identified. And among these 34 proteins, 11 were found to be associated with the prenylpropanoid biosynthetic pathway, which is the common flavonoid biosynthetic pathway. So since we know that after we adding the phenylalanines in the cell suspensions cultures, it actually can help to enhance the productions of some of the flavonoids that we are interested on. And then we also analyze the genes and also the proteomes that involve in this flavonoid biosynthetic pathway. So that is come to another questions like what next, right? So after we got those baseline data, so we look at the pathway again. So we know phenylalanine at the precursor. Then it will go through a few steps before it can actually synthesize this or produce these cyclohexanol chalcones, which is CCD. But we are not very sure because this is a well-known pathway. But the arrow with dash lines, that one is until now we are still not very clear, or the unknown biosynthetic pathway. But we look at these structures, CCD, they're having this prenyl group. So we suspect that maybe prenyl transferase, one of the enzymes that doing this catalyzed prenyl group into the flavonoids, might be involved in this step. So like, okay, so maybe we should narrow down to look at this frenulum transferase. So again, there's another step that I would like to mention, another approach. Actually, we can do is increase in enzyme activity by overexpress certain genes involved in this particular step, which is in this case, we are looking at frenulum transferase. Okay. So, okay, we have Yvonne here, my PhD students who are actually working on this particular works. So she, using the RACE method, she isolated the full-length cDNA of frenulum transferase. In short, I call it PT. So this PT, she isolated two. One is PT1, the other one is PT2. So she managed to get the full-length cDNA of these two PT2. And then when compare with others PT sequence, so we found that there are two conserved active sites containing these aspartic acid residues. So this is a typical active site for the frenulum transferase group, and both PT1 and 2 also have showing these two active sites. So if we then we predicted this PT1 and PT2, actually they have 9 transmembrane alpha helix in their structures, which means this is membrane bound proteins. So, and both also have putative chloroplast transit peptide at its N terminals here. So this is just now what I mentioned, the two active sites here. So in the loop 2 and also loop 6. So this is how the PTs look like. So this is the example for PT2. Then we run, we construct the phylogenetic tree, and then we found that actually these PT1 and PT2 are quite divergent. So they are separate far from each others. And we decided to take BRPT2 to continue for our subsequent experiment, because we only have one student to do this. Then she started to clone the cDNA of BRPT2s into different plasmid. So in this case, we are using 3 different bacteria expressions plasmids. So why bacteria? Because we think that, if we want to have a quick understanding for this particular PT2, so bacteria system is the best because it is very fast growing. So we can quickly clone, put the genes inside the bacteria to see whether this bacteria can help us to produce the protein that we want. So we constructed using 3 different plasmid, and then we also transform each plasmid into different E. coli strains, and then see how this bacteria can perform or produce the PT2 for us. So we run the PCR analysis to confirm, yes, the PT2 genes was successfully transformed and integrated into the E. coli. And then we extract the proteins, and then we check using the Western blot after purified. Unfortunately, we can't detect any PT2 proteins in this case. So then it's from us, maybe this bacterial system is not suitable because, as we predicted, our PT2's proteins belongs to membrane-bound protein. So bacteria maybe is not a very suitable system for us to produce any membrane-bound proteins. So in this case, PT2. So maybe we can switch to another system, which we straight away using a plant model system. In this case, we use the tobacco. So because tobacco is also one of the plants model system, which can grow very fast compared to B. rotunda. So so we want to save time, so we did the shortcut. So we clone the genes, PT2 genes into this plasmid, and then we transform using agrobacterium transformations into tobacco leaf. So this is what we call the agro-infiltrations. So after 3 days, we harvest the leaf, and then we extract the proteins. Then we run the Western blots analysis again. Unfortunately, we still can't detect any the protein's interest using the Western blot, then we still can detect the positive band for our positive control. So we are quite frustrated because we tried the bacteria system and then we tried the tobacco model plants and both didn't work. Then we came across with this new method, which is called pure system. So this pure system is cell-free synthesis of any proteins. So you can actually synthesize your proteins using this commercial kit, and just a few hours incubations, actually you can get your proteins out. So in this case, you do not need any system like bacteria, yeast, or any plant system. You can just synthesize in vitro. So what you need is you need to have a plasmid. Of course, your plasmids must have the gene's interest in your construct, and then of course you have to mix certain reagents in one reactions. In this case, those listed here, but because our proteins is membrane proteins, so we need a liposome here. So if your proteins is soluble proteins and not the membrane bound proteins, then you do not need this liposome. This is act it as a synthetic membrane. So after incubations, purifies, you can actually get your products very quickly. Then you can just run Western blot to check whether you can get your proteins from in vitro. So we done that. So then we check using the Western blot. After purified, we also can get our proteins out finally. So in this case, we detected the 43 kilodaltons proteins interest using the Western blot after purifications. So next, of course, since we have synthesized the proteins, the PT2 proteins here, so we will keep these PT2 proteins for our in vitro enzyme assay to check what is the really functions for this. But before I go into the in vitro enzyme assay, we actually also try to express these PTs using B rotunda cells. So just now we try bacteria, we try tobacco, and then it failed. Then we synthesize it. Yes, we get the synthetic PT2. So we think that, okay, maybe, after trying different living organisms, so I think we still have to stick into, the B rotunda cell suspensions. So we actually clone it, the BRPT2s into a plasmid, and then we transform into B rotunda cell suspensions cultures using agrobacterium mediated transformations method. Then we run the PCR confirmations. Yes, we can successfully detect the size, the PCR products with the expected size that we want in this case. So we run the Western blot. Yes, in this case, we also successfully detected the BRPT2s from these B rotunda cells. So now we have a two system here. So one is, synthesize the BRPT2 from cell-free system. The other one's PT2s extracted from B rotunda. This is transgenics, okay, transgenic cell suspension where we introduce this PT2 into the B rotunda. Then we think that, okay, so now we have two different PT2, although they are the same, but they produce from a different system. Maybe we can check the functionality of these proteins. So we run the in vitro enzyme assay. Of course, there are a few combinations of substrates and conditions here. Then we will check. Then we also think that, okay, since we have a transgenic B rotunda cells, so maybe we also can fit a various substrate straight away into the cell suspension cultures. Then we will harvest the cell and then to check whether is there any flavonoids compound can be enhanced in this case. So let's look at the in vitro enzyme assay first. So again, just to remind you, we have two BRPT2 here. So for in vitro enzyme assay, we try different type of substrate. because we afraid the PTs might be quite specific. So we try a different types of substrates. So some of them are reported before in others papers. So some are quite similar structures. So we run after we done the in vitro assay, then we have extract everything and run HPLC. Sorry, in this case LCMS. So we only detected pinostrobin chalcone and catamonins, these two substrates, and can give the product pinostrobin and also apinitins respectively. The rest we can't detect any others products from the LCMS. Then still in vitro enzyme assay. So we optimize the conditions and different substrates and also different reagents in both cases. So in this left-hand side, we have a synthesized BRPT2. So since we can manage to get pinostrobin chalcone to convert into the product and catamonin, then we just narrow down, take one, the pinostrobin chalcone to continue. In this case, then we optimize the different reagents and also the conditions here. Then, so if you can see the yellow bar here highlighted in box, these two, why they are too low? Because they do not have any synthetic BRPT2. So that's why they do not have this enzyme in vitro assay. So you can see they have a very low amounts compared to those who the tubes that have BRPT2 inside. Then we also test the recombinant BRPT2. This is the BRPT2 produced from transgenic B rotunda cell. Then again, they also showing the same under the same conditions, the S1 and S3, also without the enzyme, the PT2 enzyme. So without the enzyme, actually these reactions cannot really happen. So that's why they have a very low or minimum amounts compared to others, reactions. Okay, so we finished the in vitro enzyme assay. So these two BRPT2s actually working. Then let's look at the in vivo biotransformations. In this case, we have a transgenic cell suspensions. Transgenic means these cells, they actually have a BRPT2 genes that we introduce. Okay, then in this case, we just feed various substrates into the media so that hopefully the enzyme inside the B rotundas can actually catalyze or convert the substrate into the product that we want. So from the results, so yes, we can see the transgenic B rotunda cells actually can convert some of the substrates that we feed into. So they have a highest amount of pinosambrins, apinitins, naringenes, and pinostrobins compared to their respective untreated cell or the control cell. But unfortunately, pandroitin A and catamonins are not much difference, especially for pandroitin A. This is quite frustrated because we actually want to see the CCD product, which is the pandroitin A or 4-hydroxyl pandroitin A. But after we get the transgenic product, we feed the substrate, it still can't really enhance any pandroitin A. But it do enhance other flavonoids compound. So in conclusions, there are many works need to be done either in vitro and in vivo because we are not really fully understand the whole mechanism, how these things work. So we have another new student, Alicia Ng. So she's going to continue from there. Hopefully we can get something from there. Then there's another strategy called gene silencing. So gene silencing means if you actually want to get the product number 3, but actually this product is not the end product and it can be converted into product number 4. So in this case, we actually can just block the biosynthetic pathway here so that this thing cannot happen and we can finally get quite a high amount of this product 3 here. So this is what we call silencing. So if you have a pathway like have a lot of branching, so you can actually block the branch pathway so that it can concentrate to produce or to go to the pathway that you want. So again, we look back to this flavonoid biosynthetic pathway, and we know that you see from cinnamic acids, actually it has two branches here. One is go through the 4-CL enzyme, then it will be converted into cinnamide-CoA. And if you have C4H, then it actually can convert cinnamic acid into p-commonic acids. So we see, so our CCD is almost towards these directions, the 4-CL site. So we hopefully maybe we can just silence this branching pathway so that everything can go into this pathway so that we can get more CCD in this case. So, so to do that, we actually using RNAi approaches, the RNAi interference. So we isolated this C4H, the partial C4H, and then we clone it into an RNAi vector, which called panda vector. Of course, after we cloning, then we do a sequencing to check whether those genes, everything's in the plasmid or not. So then after plasmid constructions, we transform this C4H plasmid containing plasmid containing C4H genes into the B rotunda cell. Then we confirm that all are positive transformants. We run the Southern blot to confirm. So we have a line, transgenic line number 8. After testing with the Southern blot, it's actually positive. Then we use this transgenic line number 8 to do the analysis. So we feed them with various substrates. Then we check using this LCMS. This is the primary metabolites. So most of the compound actually generally higher compared to the control. But primary metabolite is not our interest. Our main interest is secondary metabolites, which is the flavonoid compound. Then unfortunately, we only managed to test a few compounds. In this case, we have a cinnamic acid and commaric acids to check under these two. So from blue color bars is the cinnamic acid here. So after silencing in this transgenic lines number 8, it's not much difference compared to the control, especially for the commaric acids, which means silence or not silence is actually not really significant difference. But the difference would be the very end product here, the apinitin and also the pinostrobins. So when you silence, you actually can silence this pathway. So you actually get less apinitin and also the pinostrobins compound. But this is still not our targets because students, take some time to finish the study. So and then we have a lot of issue for running this. So then the student already graduate. So we didn't really continue. So we have another new students called Carlsons who come in and continue from this work. So he will be using 2 approaches. One is again the RNAi approach. The other one is the CRISPR-Cas9. So in short, it also can known as CRISPR-I. So if you know the genome editing tool, the CRISPR-Cas9 system is actually help to edit. So it can break the double-stranded DNA nucleotide sequence. Then the genes cannot be functions, or it can delete certain genes, or it can introduce certain nucleotide into your sequence. But in this case, we are not introducing or we are not editing any nucleotide sequence. We are using CRISPR-Cas9. Okay, there are slightly different approach compared to a genome editing tool. So our Cas9 actually there's one component here called repressors. So when we have this repressor attached to the Cas9, then this whole Cas9 become deactivated. So it cannot function to cut anything. So we designed the guide RNAs to incorporate into this to form a complex So this guide RNA is actually targeting our genes of interest. Okay, so when we have a guide RNA here and combined with the D-Cas9 proteins here, so these things will go and to look for the gene sequence that we want to target. When they found the gene sequence in the genome, then it will deactivate the expressions of that particular genes. So to do that, or to increase the efficiency for this D-Cas9 system, we designed 3 different guide RNA, and then each guide RNA then later will be cloning into at the ends we have one cassette containing 3 guide RNA. Then after we done the CRISPR-I construct, then we transform into B rotunda cell. We also transform into tobacco cells. Because of the COVID, so many of our B rotunda cells was gone because no one can enter the lab to maintain the cell. So we lost many of the B rotunda cells. So that's why we will have these tobacco cell suspensions, because they grow even faster compared to the B rotunda cells. So that's why my students are initiated 2. One is B rotunda, one is tobacco. So we transform the CRISPR-I plasmid into these 2 systems. Then we check. Then we check whether they are positive. So these transgenic lines we actually harvested, and then we check their transformation efficiency. So we found that okay, CRISPR-I's actually have a higher positive percentage in transformations. Then we harvest those cells to do to have a quick analysis of the genes C4H and also 4CL. So if you still remember the pathway, these 2 are actually at the branch pathway. One is 4CL, the other one is C4H. So the one we are doing is the C4H, but we also check whether this gene nearby, whether they are upregulated or not. So after we silence these genes, so these genes, the expressions were much more lower, so it become downregulated. But compared to the 4CL, so these genes, the expressions were upregulated. Okay, so in both RNA-I and also the CRISPR-I system. So both systems work perfectly in this case. Then we are thinking like, okay, so of course, this is still ongoing work. Students haven't finished. By right, he has to extract the flavonoids and to do the LCMS analysis. But because of the lockdown, student cannot enter the lab, so he can't do any analysis so far. So at the moment, he is just stopping here. So we have another student called William. So he is a PhD candidate who are looking at the MIP transition factor genes. So just now is 1 gene by 1 gene. So we think that maybe the whole system is not really that simple. It can be quite complex. So why not we just look at the transition factor genes at this level. So transition factor genes is one of the genes that can regulate the downstream genes. So when plant under stress, those transition factor genes, they will be first expressed. So when they express, they transcribe, then they can go and regulate the downstream target genes. So we think that maybe it is a good strategy that we look at the transition factors level. So we have 3 potentials, MIP 1, 2, 3. Then in this case, we run those characterizations and predictions while doing some experiments. Then at the end, we decided to just go for MIP2 because this is the most promising MIP, and based on the previous literatures, is also involved in the prenylpropanoid pathway. So we transform them this MIP2 into tobacco plants. Okay, again, tobacco plants grow faster, so that's why we use the tobacco as a system. So we transform this MIP2 into a tobacco plant. We have a control growing without antibiotics, any antibiotics in the media, so it grows perfectly. So, and we also have a control with antibiotics media, which the antibiotic can kill the plant because they do not have a MIP2 and also the resistant genes inside the construct. So that's why it can kill the plant. So we also have the empty vector. Empty vector means the plasmid without the gene of interest. So this is serve as some kind of a control. So we have a plasmid with the in this case it's a MIP2. So MIP2 transformants. So then you can see some of the shoots are coming up after a few weeks here. So we harvest the plant and then we run the PCR to confirm, yes, they are positive. This is from the hygromycin resistant genes. So this is from the genes of interest. So some are really positive. So we are quite excited. So we are trying to get the T1 and T2 plants and to do a further analysis. So in this case, also the same, we haven't managed to get the flavonoids analysis done because of the COVID, but we do manage to finish qPCR. So this is the biosynthetic pathway again, the simplified versions. From phenylalanines, it go until CCD, so it will involve these few steps. So we check the genes PAL, 4CL, and also CHS to check whether in the transgenic plants whether they can have higher expressions compared to the control. So yes, in this case we have 3 transgenic lines, M1, M3, and M7. So all are showing higher expressions compared to the control. So this is overexpressed. So by right, it have a higher expressions compared to the control. So in short, plant metabolics engineering is really full of promise because you will really need a lot of using different kind of approaches or strategies for you to enhance or to understand certain biosynthetic pathway. But the success rate are very, very low and it's very rare. So you may need to spend a few years or maybe up to 10 years in order to just study one thing. So you see just now what we presented, this is still ongoing work and we still haven't really reached there yet. So this mainly because of a few major challenges. So because we do not really have enough knowledge on this biosynthetic pathway, and then sometimes when identifying or sourcing the genes can be quite challenging as well. And then after you identify the genes, then you have to express those genes to produce a functional enzyme. Yes, you probably can express the genes, but in order to get the functional proteins or enzymes, that also is another story. So, so that will be the main challenges that in metabolics engineering works. But of course, we still quite optimistic because with the advanced technologies nowadays, so we can actually use various engineering strategies to bring us closer to develop this the scalable process for flavonoids. So of course, this works. I have to thank many people here. So we have a plant biotechnologies group. We have a Prof. Nazranis, my mentor, and then we have a chemist, our collaborators, Prof. Nosada. We have those former and current students. Some already graduated, like Yvonne, who are working on the pre-transferase genes, and Carlson's who are still working, and Alicia, who is going to take over, just joined us just a few months ago. So with that, thank you very much. If you have any questions, yes, I'm happy to take any questions.
Thank you very much for the exciting talk, Dr. Boon. It's very exciting to see that application of genomics and genome engineering and genetic engineering in tropical plants, especially in medicinal plants. Because mostly we see this thing in Arabidopsis and then maize and then rice. It's very rare to see this kind of state-of-the-art technology applied in medicinal plants. And I hope that I can collaborate with you. We can collaborate with you in this topic in the future. Thank you very much. So now we have the discussion sessions. So we have quite a lot of questions actually from the participant. So I will write one question for Dr. Nani and one question for Dr. Boon. and we go through them one by one. So first question for Dr. Nani, it's came from Nabila Hapsari from Universitas Airlangga. The question is, I'm interested with a Dipto-Indonesian compound, maybe because the name is quite unique. Can you elaborate more about the compound? Thank you.
Okay, thank you, Dr. Anjar. Thank you, Nabila, for the question. Dipto-Indonesian A is one of our new compound that already isolate from Sorea seminis. So why we give name the Dipto-Indonesian A? Because if we get the new compound, that we can give the name of the compound like we have the children that we prepare the name of the compound. At the time, this compound is come from Dipterocarpaceae family, and then we get the sample from Kalimantan, so we give the name Dipto-Indonesian A. So why A? Because this is the first time that we isolate the secondary metabolite compound, especially on oligomer stilbene from Dipterocarpaceae. So we give A. After that, we also found another compound that we give Dipto-Indonesian B or something like that. So this is the name is very interesting, and the activity of this compound also very active. This compound is like epsilon viniferin, the dimer of stilbenoid with the C-glycoside in C12. That's, I think, if you want to see more about our Dipto-Indonesian A, you can see our publication, and then you can check in our website. Thank you.
Thank you very much, Dr. Nani. So Nabila, I hope that's answering your questions. So then questions for Dr. Boon is from Dr. Almando Giraldi from our research group. So the question is, do you think about microbial or enzymatic transformation to produce the ginger active compound? Can you suggest what kind of enzyme can be used for the biotransformation to produce ginger active compound? How is your strategy yield and cost effectiveness compared to biotransformation strategy? So I think it's more question if you expressing it in microbes, the enzyme, and can you express the final product in microbes, whether it's possible or you have to still transform it to plants?
Okay, thank you for the question. So actually we can use purely just a microbial system to produce the compound, to produce the proteins that you want. So what you can do is you insert the genes of interest that can produce the enzyme or the proteins that you want into the bacteria. So the bacteria actually is successfully expressed, then it actually can produce the proteins that you want in the bacteria system. So in this case, you do not really need to do the biotransformations in plant, but you can do it in purely in a microbial system. So but why in this case we are trying to do a microbe and also the plant system is because we found that our proteins is a membrane-bound proteins, which is very challenging to express. So it's not like soluble proteins where you can use a microbe system or you can use the yeast system to express easily. So because we want to do the shortcut first, right, the fastest. So that's why we try the E. coli first, but somehow it's not working. So that's why we have to change into the plant system to do the biotransformations for that case. Yes, you also can use biotransformations, for example, using the plant's material. So you insert your genes of interest into the plant material. Then from there, you just feed the substrate. Then by right, theoretically, you can get the product that you want. So the product from plants materials or plant tissue will be much more higher compared to microbe. Unless your microbe, you are already have an optimized system, optimize all the conditions, and then already scale up to a few liters fermenter. So once you harvest, then you can get a sufficient amount. But if you compare to the plant system, you also can use the bioreactors and they can produce much more compared to the bacteria. So what I would suggest is you really need to understand your genes of interest or the proteins of interest that you want to target first. So if this is soluble, so I think any system also can. But if this is membrane bound, either you can use a yeast or plant system.
Thank you, Dr. Pun. Dr. Armando, is that answering your questions?
Yes, thank you very much, Dr. Tan.
Okay, thank you. So now we back to Dr. Nani for the second questions. It's from Johannes from Airlangga University. The question is, if someone want to characterize total metabolite diversity instead of isolation of one specific compound, which extraction and separation technique will be the best for tropical medicinal plant?
Okay, yeah. Yeah, thank you, Johannes Kartijo, for the question. For the total metabolite, this is the secondary metabolite, I think, because I don't have experience about the primary metabolite. So for the secondary metabolite, that usually if we want to get all of the compound from the sample, that's from the plant or something like that, we have to isolate with the universal solvent like ethanol or methanol. And then after that, we do checking with TLC. But if we have the methanol fraction or methanol extract, that is difficult to get the TLC in good result. So we have to separate non-polar, semi-polar, and polar compound. And after that, we have to check one by one the TLC of them. What kind of the solvent that we have to use, that's depend on the sample that we are checking. So for non-polar compound, we checking using non-polar solvent. As you see in the video, that the non-polar will like with the non-polar. So if we have the extract, some extract of N-hexane, we usually use N-hexane dichloromethan or N-hexane ethyl acetate or N-hexane chloroform to checking the TLC to know how many compounds in that place. So in that extract. So for the semi-polar compound, we usually also use the semi-polar solvent to check it the TLC. Also, when we want to isolate the compound or extraction the compound depend on what kind of compound that we will our target. That if our target is semi-polar compound, we have to use the semi-polar solvent. That in our organic lab, we usually say that like dissolve like, as a friend that we want to talk for a long time with our friend that we like it. But if we don't like that, like in chemical also, if the solvent non-polar, they will soluble with non-polar or something like that. So depend on the target that we will extract or we will separate. Thank you.
Thank you very much, Dr. Nani. I hope that's answering your question, Johannes. And then we back to Dr. Bun. So the second question is from Nur Hayati from University of Diponegoro, Biotechnology Department. I'm not quite sure about the meaning of the question, but I will read it anyway. Would you like to give the methods to analyze new product from plants? Maybe if Ibu Nur Hayati is here, you can clarify the questions. Ms. Nur Hayati or Ibu Nur Hayati, are you here with us? I don't know if Dr. Bun want to answer that because I'm not quite sure what it's.
Okay, maybe I try to answer. I guess she's asking what are the methods that to analyze the plant's products, right? So in this case, there are a few methods that we can do. Like, you know, we know that plants tissues, they have a lot of primaries and metabolites. They also have a lot of secondary metabolites. So the difference between primary and secondaries, both are important, but primary will be more on, you know, the essentials like glucose, sucrose, all those metabolisms or the energies. But for secondary metabolites, they are also important, especially for plants' defense mechanism and also for pigmentations and all those, or anthocyanins, those are involved in the pigmentations. So in order to extract those secondary metabolites, in this case, yes, you can just using methanols and all those methods very commonly used. You can just extract all the compounds and then evaporate them. And then after that, you can use either HPLC if your compounds is can be in a liquid form. So you can use the HPLC to analyze, to detect your compound, but you need to have a standard in this case so that you can identify what are those compounds. Otherwise, you will just see the peak at a certain retention time, but you do not know what is this compound. But if you already know what you want to look at, so you probably can buy a standard, exactly the standard, so that you can compare the retention time to identify the compound. Then another way to do it is to using the LCMS. So LCMS is actually, they have a mass spec. So it's the same concept with HPLC. So you just inject into the LCMS analysis the instruments, then they will detect all those compounds. Then it connected to the database. So from there, you actually can identify what are the potential compounds present in your extract. So that is main techniques or instruments or methods that we use to analyze the plant secondary metabolites.
Thank you, Dr. Bun, for the explanation. So now we back to Dr. Nani. So for Dr. Nani, the third question is from Bu Kusuma from our research group. The question is, I just wonder how you select the solvent during your isolation of the compound from any kind of plants. Could you elaborate more about the isolation of the specific compound? So I guess this is in the TLC, and then you have several pen, and then how to isolate that one specific compound.
Okay, thank you, Dr. Anjar. I think before Dr. Kusuma, I think there is a question from Samsusu Aipu. That's which one is more accurate in separation compound? Okay, I will answer the two questions in the same time. Yeah. Okay.
Sure. Thank you.
Yeah. For which one accurate? I think that the sophisticated measurement is more accurate, I think. But I told you before that if you don't have the sophisticated equipment in your lab, it means you cannot work anything, but you still can work to get the secondary metabolite compound. Yeah. Okay. Ibu Dwi Kusuma, that's what kind of how to select the solvent depend on the compound that you want to isolate. So if we want to get the non-polar compound, I think if we want to isolate the compound, we already have the target. But if you want to isolate all of the compound, so we can start from non-polar, semi-polar, and polar solvent. That we will find all of the compound. But if you already get the target, like flavonoid, then it means that flavonoid without glycoside, the aglycone, we can find in the semi-polar solvent. So we use the semi-polar solvent. So, but in the extraction process, we need to isolate all of the compound, and after that remove the non-polar compound using the non-polar solvent. Then after that, we separate using the semi-polar compound, semi-polar solvent. And then in this extract in semi-polar, we will find the flavonoid compound. So the answer is depend on the compound, the target of the compound. Thank you.
Okay, Dr. Anik, thank you. I hope that's answering your questions. So if you have follow-up question or if you need to clarify anything, please you can just turn on your microphone to clarify it. Thank you. So now we back to Dr. Bun. This is question from Eli Triwijayanti. I'm interested in transcriptomic and proteomic methods, and this is something new for me. What are the difference in the protein of each method? Do you need some clarification on this, maybe, Dr. Bun?
I'm not sure why. What are the proteins difference?
Yeah, Ellie, if you run, maybe you can clarify the questions. Or maybe about the protein extraction for proteomic analysis, maybe like how you handle the extraction for a proteome analysis.
Okay, so as I mentioned, in this case, we are using the gel-based proteomics approach because that was 2010. So at that time, the gel-free methods is still not that common. So nowadays, if you look at the recent papers on proteomics, those they are using the gel-free because you can just easily extract your proteins and straight away inject into the LC-MS MS easily, so they can analyze those proteins. But if you go through the gel-based, then you have to run the two-dimensional gels electrophoresis first to separate them. So the first dimensional gel electrophoresis will be separate according to their PI value. You know, each proteins, they have a different PI value. So when they separate through the first dimensions, then the protein will move according to their PI value until the PI value becomes zero, the net zero. Then it will stop there. Then after we finish the first dimension, then we continue for the second dimensions. So gel electrophoresis. The second dimensions will separate the proteins according to their molecular weight. So just now is PI value. Then the second one is molecular weight. So the protein will move against according to their molecular size. So the biggest protein size, they will move very slow. So that's why you will see they are at the top of the gel. So those are very small proteins, they can move very fast. So that's why you will see those proteins are actually at the bottom of the gel. So in this case, you already separate according to first dimension and second dimensions, then you will clearly see those proteins become a spot, you know, a round shaped spot. So from there, you compare with your control and also the treatment gel. These two gel, after you compare, of course, with the help of a software, using a naked eyes will be really, really tough. So after you compare, then you will identify what are the proteins actually have a different expressions from there. Of course, this one you have to use the software. Then some protein missing in the control, but some proteins appears in the treatments gel. So you will select those proteins which have a differentially expressed or missing proteins, and then you will send for LC-MS analysis. So in the LC-MS analysis levels, then they will inject one by one the proteins so that you can identify each protein, what is these proteins are. Then this is proteomics. So of course, nowadays, we have a gel-free technology, which you extract the total protein, you put the enzyme to digest them, become a peptide, so that it can easily inject into LC-MS. Then they can analyze everything for you. So from there, of course, you search through the database, you can easily identify the proteins. So that's why this is much more faster and simpler. So recently, those papers are looking at the gel-free. Then for transcriptomics, so transcriptomics also have two ways. One is a microarray, and then the other one is RNA sequencing. But of course, nowadays, many people are using RNA sequencing because it can be very cheap compared to the microarray. Microarrays, you need to have all those probe that potential probe that can target your genes, certain genes. So the company have to design all those probe and then put it in chips and then into a valve. So you put your sample there. But RNA sequencing is like NGS, so it can just extract RNA and then you just inject into the machines. Then they can analyze or sequence all those. Then after that, you have a lot of data. You have to do the annotations, everything to analyze your data. So that is the difference between transcriptomics. So transcriptomics is targeting the RNA level. For proteomics, is targeting at the proteins level. I hope I answer your questions.
Thank you, Dr. Bun, for the explanation. As I mentioned before, if you have any follow-up, you can turn on your microphone and ask for it. Okay, if there is no follow-up, then we go to the next question for Dr. Nani. It's from Umi Zubaida. The question is, you have mentioned that during TLC, you need a specific strategy. What kind of strategy that you mean? Could you give more example and explanation about the strategy?
Okay. May I share my slide, Dr. Anjar, to answer this question?
Yes, sure, yes.
Okay. Can you see my slide?
Yes, we can see in the slide.
Yeah. Umi, thank you for the question. The strategy is before we separating using vacuum liquid chromatography or column chromatography gravitation, we have to check the TLC first. For this extract that we try to find the TLC many ratio of the eluent, many ratio of the solvent that you can see here. If I use the hexane and dichloromethan 9,1, that all of the compound stays in here, and then only one compound move. So if we do this solvent for the separating using vacuum liquid chromatography and or gravity column chromatography, we will find this compound. But if we use this eluent, this hexane dichloromethan 3.7, that we not only get this compound, but we will also get 1, 2, 3, 4 compound in the same time that they go down. So the strategy, it means that we have to find what kind of the solvent that good in the TLC, and then after that, we use it in column chromatography, vacuum liquid chromatography, or gravity column chromatography. If we don't have the good TLC, try again until we can find the good separation in chromatography. If we see like this, there are 1, 2, 3, 4, 5, 6 compounds. So this type of TLC is very good to check the kind of compound that already come in vacuum liquid chromatography or gravity column chromatography. But if we use this one for the column chromatography as solvent, that all of the compound will go down together. It means that the strategy to get the solvent, the good solvent in separating process is very important. So usually we separate the sample by increasing the gradient of the polarity of the solvent. I think like that, Dr. Anjar. Thank you.
Thank you, Dr. Nani, for the explanation. we have several questions, but I think because time limitation, I think will be the last questions for today. So I apologize for the unanswered question. You can contact Dr. Nani and Dr. Boon via email if you want to elaborate further or ask further with Dr. Nani and Dr. Boon. So will be the last questions for today for Dr. Boon. The question is, thank you from the explanation for different method of increasing metabolite productivity in Zingiberaceae in ginger. Which, based on your experience, which method will be the most effective, suitable to be applied in industry? So you explain about silencing and then overexpression and then so on. Like based on your experience, which one is the most effective to increase the metabolite and will be the most suitable to be applied further in industry? Thank you, Dr. Boon.
Okay, so thank you for the questions. Actually, if you want to apply or extract those compounds to be applicable for industry applications, actually you can just straight away use the rhizome. So you just extract all those compounds from rhizome. Then you actually can get a lot of metabolites from rhizome directly. But because we have these problems where if you use a different batch of different seasons of the rhizome of this B. rotunda, actually it will affect not only for the plant's growth, but it's also for the metabolites compounds. So that's why we want to be more consistent. Everything's under a controlled environments and conditions so that we can produce throughout the whole year. So the best systems in the lab actually will be the plant system itself. So if you can manage to establish the self-suspensions cultures, actually you can use the bioreactor to scale up from 3 liters to 5 liters or even 10 liters if you want. So from there, actually you can produce a lot of cells, and these cells actually can produce a lot of secondary metabolites. So another strategies you can use, which is quite easy to do, instead of go through this genetic engineering, which is using the elicitations. So you can use the elicitors to add into your media so that it will create the stress to your cell. And then when the plant cell under stress, they will release a lot of secondary metabolites. So this is the functions of secondary metabolites. When under the stress conditions, the plant cell will release a lot of secondary metabolites. So you can actually use this way, and with the help of bioreactors, you can produce a lot of secondary metabolites that you want. But of course, genetic engineering also possible, but it will be much more tedious because you need to, introduce certain genes into your plant system. So it's not 100% success. Even the transformation rate also very low. Then whether these the genes can be really functional at the end as a product. So that's why for us, we are interested to understand the biosynthetic pathway as well, besides just producing the secondary metabolites. So we use the different approaches, including RNAi, CRISPR-I, and also some of the conventional overexpressions method to enhance our targeted secondary metabolites. So the most easiest and more applicable for the industry would be like, the bioreactors for the plant cells. That will be the much more easier. But of course, you also can use the bacteria system, as I mentioned. If your proteins is not membrane-bound proteins, you can actually definitely go for bacteria and also the yeast system. So that one also works. But of course, you have to check your proteins, whether at the end, the proteins that produce, is it functional or not. I would not recommend you use the in vitro. That one is just for testing in the lab. In vitro will be really, really expensive. It can, just now I show you the pure system. So that one is a kit. 10 reactions, it will cost us about 1,000 USD. just for 10 reactions. And at the end, you only get about 10 microgram of proteins for the whole, at the end, the reaction. So this is way much more lower that you can use for industry. So that one is just for lab testing purposes.
Thank you, Dr. Boon, for the thorough explanation. So we come to thank you so much for our speakers. So let us give our huge appreciation for both of the speakers. Thank you for the effort and thank you for the time for sharing your work and research with us today. So before I close the seminar, we have a photo session for all of us. So Dr. Almando, can you please coordinate the photo session?
Okay, thank you, Dr. Anjar. Okay, everyone, please turn on your camera and give your best smile for the photo session. Okay, so there is 8 pages in this Zoom. So I will start first with the first page. Okay, 1, 2, 3. Okay. Okay, then I move to the second page. Okay. 1, 2, 3. Thank you. And then, oh, start from third page. Nobody turn on the camera. How is it, Dr. Anjar? Do I still need to take the photo?
Yeah, yeah. Okay. Yeah, it would be good to take the pictures for documentation.
Okay. Okay, 1, 2, 3. So then, please wait for me. I will take another 5 page. Okay, 6 page. Yeah, 6 page. Seven page. And the last one. Page. Okay, it's finished. Thank you, everyone. And I give back to Dr. Anjar.
Great. Thank you, Dr. Almando. So before I close the seminar, I would like to remind the participant to fill out the certificate form. We will send you the seminar certificate to your email. Please wait 1 to 3 days. It will take us 1 to 3 days to send the certificate. If you cannot find it in your email, please check your spam folder because sometimes it goes to the spam folder. And so thank you for a huge appreciation for both of our speakers. Thank you for sharing your research and your work with us today. And then some participant asked, can they get the PowerPoint of your presentation? So Dr. Nani already shared his presentation. I don't know if Dr. Boon will be willing to share your PDF of your presentation to the participant.
Yeah, sure, no problem.
Recording stopped.
I will send to Prof. Wulan.
Recording in progress.
Okay, thank you, Dr. Boon. If you can send the PDF to Professor Wulan, then I can share it to the participant. Thank you very much.
Yeah, sure.
Yeah. Okay. All right. So we are at the end of the seminar. So this seminar is aimed to share and discuss research and finding, especially in the field of tropical plant biotechnology. We would appreciate if you can check our website and drop us email if you want to start a collaboration with our research group. And of course, for the unanswered question and further question, you can also email Dr. Nani and Dr. Boon for potential collaboration as well with their lab. So thank you, everyone. we are still in the middle of the pandemic, but it really great that we can come together here and still productive, still can sharing our research and our work to each other. So thank you for being here today. Stay healthy, please. Follow the instruction for the government. Wash your hand, wear the mask, and keep the distance between each other. Thank you very much, everyone, for being here. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon and have a good weekend.
Waalaikumsalam. Thank
Thank you very much.
Thank you.
Prof.
Thank you, Dr.
Ulan.
Dilan. Terima kasih.
Prof.
Terima kasih, Bu Nani.
Ya, Bu Nani.
Terima kasih, Pak Anjar.
Dr.
Terima
Pun,
kasih.
thank you, Dr. Anjar.
Thank you.
Terima kasih, Bu Nani. Terima
Thank
kasih
you very
semuanya.
much.
Bu Nani, presentasinya di email ya?
Tadi saya email Pak Anjar.
Ya, nanti saya bagikan melalui email ke
Di
peserta.
PDF kan saja mungkin ya?
Ya, di PDF kan dulu, monggo Bapak Ibu kalau
sudah ya.
Kalau mau lah.
Bu Wulan sudah enggak ada.
Terima kasih Pak Gary, terima kasih Bu
Terima kasih semua.
Terima kasih semuanya.
Salam sehat ya.
Ajar, sehat selalu.
Maksimal.
Assalamualaikum.
Assalamualaikum warahmatullahi wabarakatuh.